Hello, Dr. Grismer. Thank you for joining me today on
Sabbatical, Spotlight Series, Sabbatical, Spotlight Series, Sabbatical, Spotlight Series, Sabbatical, Spotlight Series.
Sabbatical Spotlights.
Professor Grismer is the Director of Research and a professor in the biology department. He spent his spring sabbatical writing manuscripts for his research with new gecko species in Thailand, the Philippines, and Cambodia.
Tell me a little bit about your sabbatical.
My sabbatical wasn't at all what I had planned. I had planned to be in a different country every month, starting in April and ending in October. And I had all the arrangements made with all my colleagues in all these other countries, and then that completely collapsed because of COVID-19. So I made lemonade. And what I did was I got caught up on a bunch of manuscripts that we had in press that we were going to add data to with new fieldwork, but obviously we couldn't. So we finished writing up what we could with what we had. And this actually dealt with several new species that we had found in Myanmar, a new population that we'd found in an island off the coast of Cambodia, and then some really cool stuff that we got on the western edge of Thailand where it borders Myanmar and some really sort of places where not a lot of people go. Quite exciting, actually.
Can you tell me a little bit about your previous research trips and how you catch lizards?
Well, the research trips are really sort of low tech. We pull up Google satellite. We look for an area on the map that has an interesting topography, such as an isolated mountain range or something like that. And we realize that nobody's ever been there, so we don't know what's there. So let's go there. And so we make the appropriate arrangements with the appropriate people in the appropriate countries, and we get all the permits down, and we get as much money together as we can. CAS has been incredibly helpful in that regard. And then we fly to that country, hook up with our contacts, bribe the appropriate people when necessary, and then head out into the field and hope that we get lucky. And so we go out during the day, and we check out a place to look out during the day because the animals we are hunting come out at night. And so we don't want to wander around these hillsides that are covered with landmines and militia groups and stuff like that at night and not knowing where we're going. So then we go back out at night after we set a route and we catch stuff. When we're out collecting during the day, we're looking for species that live high up in the trees. And to get those down, we shoot them down blowpipes. We buy blowpipes online that are aluminum blowpipes lined with Teflon. They're 40 caliber. We buy plastic stun plugs. And then we can shoot lizards down off the tops of trees. And it's usually pretty new because they're going to places where no one's ever been or no one ever wants to go. And we photograph them. Then we take tissue samples of them, which we bring home and we generate DNA. And then from the sequences that we get out of that tissue sample, the DNA sequences, we put those into a computer with a bunch of other sequences from known species. And the computer has a mathematical algorithm which generates what's referred to as a phylogenetic tree, the tree of life, okay? And if we have what we suspect is a new species, it'll branch out in that tree of life on its own branch. In other words, it'll be the only living organism on the planet that occupies that same branch. And so if we see that, then we're pretty much assured that what we have collected is a new species. And if that's the case, then we compare its anatomy and its color pattern with some of the other closely related species on that tree, so we can define it from other species. So if someone finds one, they don't have to do a phylogenetic analysis. They can simply look at the anatomical and color pattern characteristics that we have laid out in the manuscript, and then they can determine if that is the same species that we caught, or yet if it's completely new species as well. And this is what I was working on a sabbatical, was publishing these species descriptions from previous trips. It's an interesting process. Takes quite a long time, a lot of statistics involved, a lot of careful scrutiny of morphology and things like that. There's a bit of a learning curve, but I have some of the students in my lab right now are just phenomenal at doing that. And they're making great progress and publishing their own papers as well.
How does your ecology research in regards to speciation protect and conserve our planet?
So it's really important that we catalog the biodiversity of this planet, okay? That is, if there's new species out there, it's really important that we know that they're there. And it's not so much for the fact that we need to just conserve these species for the sake of conservation, a healthy ecosystem is important for us if we are to remain healthy as well. In other words, when ecosystems collapse around the world because of degradation or whatever thing, people die because people depend on these ecosystems, okay? And so if we are to conserve the biodiversity of this planet as well as support our own human health, we need to be cognizant of that. So think about this. Go to an area, think of a place in the world right now where people are sick and dying, okay? Now we can take those people and fix them medically. We can make them well with medicine. But when we put them back in that dying environment, what's going to happen? They're going to get sick again, okay? This is not a medical problem. It is an ecological problem, okay? And to solve this problem, we need to fix the ecosystem first, then work on the medicine, okay? Then everything would pretty much, hopefully, would fall into place, all right? So, you know, by describing species and understanding how these ecosystems work, we are in essence then contributing to our own health and safety as well.
How did your interest in reptiles begin?
I honestly think I have a particular sequence of genes in my DNA that codes for be interested in reptiles, because my earliest memory as a human being was catching lizards was trying to catch lizards by my swimming pool in Corona del Mar. My mom says I was 2 years old at that time, but and I just remember seeing those lizards and I was just absolutely fascinated by them. There was nothing in the world that looked that cool and was that amazing. And even still, when I roll up into some cave that no one's ever been into and I'm half a mile underground and I see some new gecko on the side of a wall, that exact same feeling is there. And I'm just overwhelmed with excitement and passion. You just want to figure out what it is, who it's related to, and then share it with the rest of the world.
Thank you for your time, Dr. Grismer.
All right, thank you, Jillian.
We will see you all next time on
Sabbatical, Spotlight Series Sabbatical, Spotlight Series Sabbatical, Spotlight Series Sabbatical, Spotlight Series.
Sabbatical Spotlights.